Hello, everybody. Thank you so much for attending this webinar. It's always really cool to see how many people are interested in the things that we do and to be able to share some of the updates that we've had with you guys. So thank you very much for being here. So I'm Helena. I'm the communications person for Open Targets. So if you've seen anything from Open Targets, tweets or LinkedIn posts or blog posts, that's probably me. So hello, I am the I am the communications person. And the topic of today is to kind of show you how you can explore target disease associations with the Open Targets platform, and particularly with some of our new features. So we've had some updates in the past year, a whole new way of looking at associations and a target prioritization page, which I'm excited to introduce to you today. So before I jump into the platform, I thought I would introduce you to Open Targets in case you didn't know about us. And I'll do you a brief introduction on the Open Targets platform, and then we'll dive straight into a walkthrough. I'll demonstrate how it works. And you can kind of take a look at the new features that we have. I will stop for questions as we go. So if you do have a question while I explain something, please do feel free to put it in the Q&A. Any questions that we don't pick up at this point, we'll pick up at the end, as Ajay mentioned. So yeah, we'll try to answer as many questions as we can. So a brief introduction to Open Targets. So who are we? Who are we that make this platform? So here, I thought I would talk about the kind of reason for what we exist. And I think this informs a lot about how we create the platform. So it should help explain that a little bit. So the big question, so as you may know, developing a new drug is a really complicated, it's a really costly process with a very low success rate. So as new drugs go through the clinical trial pipeline, a lot of them fail at some point in the clinical trials. And so in order to get one FDA-approved medicine, you really have to put a lot of investment into this process. And at Open Targets, we started thinking about this. And when you start, the drug discovery process, when you start thinking about creating a drug for a specific disease, we thought really the first step is target identification. So picking your target, your molecule, your gene, your protein, with which you're going to try and modulate in order to affect the disease. And so this is where we wanted to have our impact. And part of the things that informed our thinking was some research that was done in terms of genetics. So we found that genetics really increases the chances of success. If you have genetic evidence that supports your link between your target and your chosen disease, then you are much more likely to have a drug that makes it through the clinical trial pipeline. So this statistic here is from a paper in 2015 by Matt Nelson. And so they showed that you were 8 times more likely to succeed if the target was identified in Mendelian genetic evidence, twice as likely to succeed if the target is supported by GWAS evidence. And then more recently, Open Targets, we did a retrospective analysis of the drugs that were approved by the FDA in 2021. We've also recently done one of the past 10 years. And basically what we see is that the majority of drugs do have human genetic evidence to support them. And so the main point of this is really that how important it is to have that evidence that supports associations between targets and diseases. And so this is how Open Targets came to exist. So Open Targets is an innovative, large-scale, multi-year, pre-competitive industry-academia partnership. What does that mean? That means we try to bridge the gap between academic research and the pharmaceutical industry. This is all pre-competitive, so our pharma partners can collaborate together, can collaborate with us on different research projects, and this is all done before kind of any issues about IP or that sort of thing. We are large-scale multi-year. This means we are very ambitious in terms of the projects that we have, and we try to, to really try and be very ambitious, try and have projects that are really going to change the drug discovery spectrum. So who are our partners? Well, one of them already, EBI, because this webinar is organized by EBI. So we have 2 academic partners, the EMBL-EBI and the Wellcome Sanger Institute. EMBL-EBI bring their kind of expertise in bioinformatics, in large data. The Wellcome Sanger Institute has amazing capabilities in genetics and genomics. And then on the industry side, we currently have these 5 partners, so GSK, BMS, Sanofi, Pfizer, and Genentech. And they bring, the industry perspective, and they bring their expertise in their respective therapy areas. And they collaborate with us on this, which means that hopefully all the research that we do can feed directly into drug discovery pipelines. So we use human genetics and genomics data to systematically identify and prioritize targets for drug discovery. What does this look like in practice? So everything that we do tries to build up this evidence between targets and diseases, and we do that kind of through two ways. So on the left, we have our research program in which we try to have new data generated that either supports that link between targets and diseases, or we also do research to create foundational data to kind of understand more about the diseases that we're trying to cure. On the other side, we have analysis of publicly available data, and this is where the Open Targets platform fits in. So we also have Open Targets genetics, which I'll not talk about today. But yes, so these two platforms, the idea of this is to take publicly available data, to analyze it, to present it to you in a way that's kind of harmonized, systematic, so you can see the data that's out there. I'm just going to stop here for questions in case anyone had any questions about Open Targets or about anything I've said so far, before we move on to talking about the Open Targets platform.
Hello, we have one question here. How much data have the commercial partners contribute?
That's a good question. So the commercial partners don't contribute any data to the platform because the data that we use in the platform is all publicly available. So we will only include data that anyone can access. Okay, I can move on to talking about the platform. So just a quick summary of what I've said so far. So target disease associations supported by genetic evidence are more likely to result in successful drugs. And so Open Targets is an industry-academia partnership that creates and applies evidence to build therapeutic hypotheses, so linking targets and diseases in these therapeutic hypotheses. And I should, so if in the summary slides, all the pictures are from the campus. So if you've ever wanted to see campus, then here's a little tour of where Open Targets lives. So the Open Targets platform. So the Open Targets platform is a data integration and analysis tool for the systematic identification and prioritization of drugs. And you'll probably hear me say that phrase quite a lot in this webinar, because that really is what we do. We try to systematically identify and prioritize drug targets. The platform is available on the web. So if you go to this URL, platform.opentargets.org, that's where the platform lives. You can explore all the data that we have. And it really is kind of a big data platform. So we integrate over 20 different data sources to build up and score evidence for target disease associations. Yep. The web interface, as I've mentioned, which you can access here. You can look up targets, you can look up diseases and phenotypes, and you can also look up information on drugs. The platform is open source, so if you'd like to take a look at the code, you can. We also make the data available through a GraphQL API, through Google BigQuery, and through data downloads. I will go through these options at the end, so if you'd like to find out more about those, stick around and I will discuss the different options you have for accessing our data. The other thing I should mention is that the platform is regularly updated, so we have 4 releases every year. And for this, we try to keep all the data that's in the platform up to date, but also as much as we can, we try to integrate new features, new data. So we're always interested to hear what data people would like to see in the platform. So if, as you're browsing, you see, you see something that you would like to see in the platform, please do let us know, because that is very, very helpful information for us. So our data model. So what does the information in the platform look like? So the first thing is targets. So we integrate information about protein and non-protein coding genes, and our source for this in the platform is Ensemble. We also integrate information on diseases and phenotypes. So these can be common or rare diseases and phenotypes, and our source for this is EFO, so the experimental factor ontology. And the final entity that we have in the platform is drugs. So these are any molecules with a known mechanism of action or an investigational or proved indication, and our source for this is Chembl. And really the power of the platform is in how we link these together. And so I've mentioned this before, so that the whole point is to build up this evidence and to show you this evidence that we have that links targets and diseases. I've mentioned, evidence a lot before. So what do I mean when I say evidence? Well, if we look here, drugs are a source of evidence, for example. So a drug will be linked to a target via its mechanism of action. It will also be linked to a disease via its indication. So if you know that you have a drug that has a mechanism of action for one target and an indication for a specific disease, that constitutes evidence that will link that target and that disease. Within the platform, we have quite a number of different sources of evidence. So I've mentioned we had over 20 different data sources. You can see most of them here. And if you're familiar with some of these, you'll see that there's, really lots of different types of evidence. There's genetic evidence, there's drug evidence, there's literature evidence. And so the power of the platform is in taking all of this evidence, all of these different sources, all of these different types, and harmonizing them and presenting them to you in a way that we hope is kind of intuitive and easy to follow. The other thing that we do is that, some of these data sources might be curated, some of them might be predicted. And so we try to really show you all that data and show you what we've done and analyze it in a way that makes sense. So I thought it would be worth me spending a little bit of time talking you through how we integrate data so you can get a sense of how this looks like in the end. So the first thing that we might do is to source new data, and any data that we integrate into the platform will fit into one of these categories. So we have genetic associations, somatic mutations, which is very relevant for oncology targets, known drugs, pathways and systems biology, RNA expression, text mining, and animal models. So if we find a new source of data, usually it will fit within one of these 7 different data sources. Once we have data, we do an ontology expansion to try and see which diseases this will apply to. And I'll go into more detail on this when I show you the platform. So I'm not going to spend too much time on it here, but it's important to note that all of the diseases within the platform are linked in this ontology, which is very important for the kind of building associations and building up evidence. So then once we know which diseases and which targets we want to associate, we can score the evidence. We can give that evidence a score, and we'll do this in a certain way. So we'll start by scoring the evidence itself. All the evidence for a data source will then be accumulated into a data source score. And then we'll aggregate all the data source scores into an overall association scoring. We'll then collect an amount of data in order to provide annotation information for our different entities. So each of our different entities, so drugs, targets, and diseases, will also have a lot of information to kind of tell you what we know about those different entities. So for example, for a drug, we'll show you all the information that we have about that drug. We also perform some prioritization. So this will be based, for example, on tractability or safety or expression for the different target disease associations we have in the platform. So that's all quite theoretical. What does that look like in practice? So here is what an associations page looks like in the platform. And this is the associations page that we introduced last year. We call it Associations on the Fly. And essentially what you have here, so we present, what I've done here is I've looked for a disease. And this page is showing you all of the targets here on the left that are associated with that disease. These will be ranked by their overall association score. And then we show you the breakdown for each data source that we have, whether we have data for that data source, and how much evidence there is from that data source. This is grouped into the different data source types that I was telling you about, data types. So we have, for example, the first chunk here is genetic association, but the 7 different data types that I told you about are all represented here in this page. The other thing that you might have noticed is that we do the scoring through a shading of blue. So the lighter the color, the lower the score, the higher the score, the darker the color. And this is a score between 0 and 1. And white cells will mean that there is no data. What's important to note here is that the score gives you a sense of the amount of evidence. So we do not provide a confidence score in the association. Rather, what is represented here is giving you a sense of what exists, what research has been done. And so the target disease associations with the highest scores will be the ones that have been the most researched. What this means is that you might have an association with a lower score that could still be very interesting, but there just hasn't been that much research done on it. And I mentioned that we release 4 times a year. So our latest release was in December 2023. Our next release will be in March. And as of December 2023, here are the kind of platform stats that we have. So we had data on over 62,000 targets, 25,000 diseases and phenotypes, 17,000 drugs. These were all connected by over 16 million evidence strings, resulting in about 8 million target disease associations. Now, there won't be a quiz. I'm not going to ask you to remember these numbers, but the point I'm trying to make here is that it's really a big data platform. There is a lot of information contained within the platform. So I hope that you find what you're looking for within that. I'm going to stop again here for questions because I think I went through that quite fast. So if there's any questions about the platform, please do let me know. After this, I'll be moving on to a walkthrough.
Do we have data from hospital samples?
Oh, that's a good question. I don't think we do. So I think within the platform, we don't have kind of clinical data unless it's coming from maybe, we have some data, for example, on once drugs have been approved, we have some data from clinics about like whether there have been any adverse reactions to that. But otherwise, I don't think so. Yeah.
Does OpenTarget provide clues for personalized medicine?
Not currently. I think this is something that we are thinking about because we're trying to, for example, within diseases, of course, disease is a huge umbrella. There's lots of different disease stages and that sort of thing. So that's something that we're trying to work towards, if that's what your question was about. But no, currently there isn't kind of like a personalized medicine section.
And the other question is about the updates. So do you do quarterly updates on platforms? Okay.
Yeah, so 4 times a year. Yeah, quarterly updates.
Right. Is it possible to focus on just one specific part of specific disease on this platform, such as mitochondria in Alzheimer's disease?
It will depend on whether it's in the ontology. So as far as I know, the ontology doesn't go that specific. You might find evidence for that within the platform if you look for it, but chances are it will probably be classed under, Alzheimer's as a whole. Yeah, that would be my.
And all the data which is on OpenTarget's platform, is it all human derived?
Yes. So all the data is from human genetics or genomics, unless it's animal model data. That's one of the data sources that we have, but otherwise it will be from human data. So for example, you will only find human targets within the platform. If you're looking at an infection, for example, you will see human targets, but you won't see bacterial targets.
And I think this is based on the demo that you gave. Can you get the specific score for the data beyond the blue shading?
Yes, you can. I'll show you what that looks like when we do the walkthrough to the platform. Give me 1 second. Okay, so this is what the homepage looks like. Let me just move the Zoom things over. There we go. So this is what the homepage looks like at the URL I gave you, so platform.opentargets.org. And so what I thought I would do is kind of try and show you what the different, how someone might explore the platform. So let's say, for example, I'm a researcher, I'm investigating a specific disease. Let's see what the platform shows me on that. So the first thing that I will do is I will look up my disease. And in this case, I've chosen to show you psoriasis. And so when you do a search, the platform will show you the top top hit for this, but you can also look at the other results that you have. In this case, I'm going to click here because this is the one that I'm interested in. And this is your associations page for psoriasis. So this is the same thing that I was showing you in the presentation. I've looked up a disease, and here on the left, I can see all of the targets that are associated with this disease. One thing that I would like to note here is that we try to include as many synonyms as we can. So for example, here at the top, you'll see that we try to show you, so this is the disease as it's called in our ontology, but we will try to show you how this relates to other ontologies as well. Very quickly, I'm just going to click over to the profile page for psoriasis. So this is the annotation data that we have for this disease. And here it's showing you the synonyms that we have for this disease within OpenTarget's platform. So let's say, for example, you were to type in psoriasis with an E, it would show you this page. And here I just wanted to talk about the ontology very quickly because I've mentioned it a lot. So this is what this looks like. So here, this is showing you where this disease that I've looked up, psoriasis, sits within our ontology. Why this is important is because if we have data that associates a child term of my disease, so let's say, for example, I have data for psoriasis vulgaris, this will show up when I look at evidence for psoriasis itself because it's a child term of the disease. This is important because it means that if you're scrolling through the platform and you see evidence that might not be exactly for the term that you've searched for, this could be a reason why. So we propagate this evidence up the ontology. So for example, let's say you would then go to, if you were to go to immune system disease, you would see evidence for any immune system disease that we have in the platform. So yes, I just wanted to explain that quickly here about what I meant by propagating up the ontology.
Okay.
Let me switch back to the associations page. And so here, what you can do, so here you see all of the data sources that we have that contribute evidence to associations between targets and diseases. And as I said here, you have the scoring. These are ranked by the overall association score. If you hover over the squares or the circles, you will see the score that we give it. So for example, here you can see that the overall score is 0.81. If I hover over these individual ones, you will see that score as well. One thing that I can see here, so I can see here at the bottom, I have 6,246 targets associated with psoriasis, so that's quite a lot. And this is only showing you 50 at a time, but you can look at all of the diseases that are all the targets that are associated with this disease. You can also sort, so say for example, these are sorted by the overall score, but say for example, you were specifically interested in one data source, you could click on it and it would sort this page through for that data source. So here it's showing you the top rated ones if I take into account open targets genetics. I'm just going to click back on association score. Great. And then the other thing that's cool about this page is that you can directly look at the evidence within this page. So let's say, for example, I want to look at ClinVar information for IL-36RN. I just clicked on the cell here and it's showing me all of the evidence that is informing this score. I can do this for so I can do this for anyone that has evidence. So here, for example, I clicked into UniProt literature. I can click into our bibliography. So this is using a text mining pipeline, bringing up all of the publications in which this target is mentioned with this disease. And so that's what that looks like. One thing I wanted to mention here as well, is that where possible, so where you see blue links, that's telling you that it's linking to somewhere. So where possible, we try to link back to the original data source. So here, for example, if you were to click here, you would go out to the original record in clinicaltrials.gov. We also have links to other pages within the platform. So for example, let's say I could click on ustekinumab. This is taking me to the ustekinumab page within the Open Targets platform. And this is showing me all of the data that I have on this specific drug. So all of the mechanism of actions, the indication for this drug, any clinical precedents. I wanted to show you here as well. So this is the pharmacovigilance widget that I was mentioning earlier. So this is from the adverse events from the FDA adverse event reporting database. So this is some of the data that we have that's from healthcare professionals, for example. I'm just going to go back to the page that I was on before. So I'm back on the associations page. And here I want to show you some of the features that are quite cool in this page. So for example, let's say you're specifically interested in one target. You can pin that target. It will appear here at the top. Let's say I also want to compare that to TNF. It means that you can now directly compare these two that you're interested in. What's also happened here is that it's modified the URL so that it remembers which ones that you've pinned. So you can save this, you can share it with other people. And that's quite a useful feature that we've developed. The other thing that I want to show you, which is very cool for this new view, is that you can actually directly control the weighting of each data source within the overall association score. So the way that we calculate the score, we take into account each of the data sources, but they are not weighted equally. So for example, let's say something like EuroPMC, which is text mining, so not a curated data source, for example, we will downweight that. Whereas something like the Genomics England panel app is full weighted because that's from a curated data source, for example. So these are just score, these are weightings that we've come up with within the team as a way of creating this score. But what you can do is you can change that score if you like. So let's say, for example, you don't want to take into account any of the evidence from gene burden, you can downweight that, and that will recalculate the score and show you the new associations. And that's why we call this view associations on the fly, because the view will directly recalculate the score as it goes. The other thing that you can do is you can require certain data sources. So again, let's say, for example, you're only interested in associations that have evidence from open targets genetics. You can require it, and that will mean that all of the associations that you see will have open targets genetics evidence. And you can see, for example, here that's narrowed it down to about 800 target disease associations. You can also click on the bar, and that will require the whole data type, for example. I'm going to reset that to default. Great. A couple of other things you can do. You can directly search for your target. So let's say, for example, you don't want to scroll through the 6,246 associations. You know exactly which one you're looking for. You can search for it in the search bar here. Let's say, for example, IL-17, and it'll bring up the different targets. A couple of other things that you can do, which I just wanted to show you here as well. You can export this data. So what's this? You've made your modifications to the score, for example. You've required your data sources. You can directly export these results from this view for further analysis. You can also export data from each of the widgets. So let's say, for example, for TYK2, I've gone into the Open Targets Genetics widget. You can download this table here. You can download it as in JSON or TSV format. And I also wanted to point out here, if you're interested in using the API, this is quite a good way to get started, because if you click here, it'll show you the API query that feeds this specific widget. So say, for example, you wanted to extract specific data, this might be a good place to get started. I think that's everything I wanted to show you for this walkthrough. So I'm going to stop here and see if there are any questions before I move on to the next section of the presentation.
Yes, we are getting many questions today. There are many questions about the data. I think I can pick a few. So one is it possible to obtain data for a certain cell type?
So we do have cell type data. So we do have some expression data, for example. You wouldn't be able to kind of filter the evidence by cell type, if that makes sense.
Can this platform be operated offline as well?
No, so you need to have access to the internet in order to access the web interface. You could download the data directly, so I will show you what this looks like later, but you can actually download these data sets onto your machine, and so you could do analyses offline.
And one good question is, can this data be used for publications or thesis? If so, how is it cited?
That's a great question. So yes, you absolutely can. We ask that you can reuse any of the data, any of the code. We just ask that if you do that, you cite us. So I'm going to switch over. This is the documentation. If you go here into licensing, you will see all the information that we have about reusing the data, reusing the code. And if you use it, please cite our latest publication, which is always linked here. That's a great question. I'm very excited that you're thinking about reusing this already.
Next is, this is a tremendously useful resource. What is your roadmap for the next few releases? It will be useful to know what is coming up.
So I can't currently share that right now. We are working on some roadmaps, which we're hoping to be able to share on the Open Targets community. In general, as I mentioned earlier, it's always about integrating, new data, that sort of thing, trying to expand what we have in the platform, trying to refine, trying to make it more granular. In general, that's our kind of general roadmap. We don't have a specific roadmap that we can share at this point, I'm afraid.
Can we look for SNPs associated with diseases?
You can't do that in the Open Targets platform. You can do that in Open Targets Genetics. So this is, if I click, will it? Yeah. So this is our kind of sister platform. It feeds into the Open Targets platform. But in here, you can look for, so you can explore the data that we have on SNPs.
One question is, how can this help NHS patients when they get genetic test reports? Can they compare it with the platform?
I mean, yeah, it's a question. I think if you were a patient, you could definitely explore the platform. We don't make the platform for patients, so there isn't a lot of explanation, about different variants, about different things. Like the data is there, but it's not like I said, there won't be a lot of explanation as to what it means or what the consequences might be. This is very much a platform that we make for scientists. But if you want to look at the data, you absolutely can.
One question is related to GWAS catalog. So you showed that GWAS catalog was a partner, but it is not shown on the panel. Do you know if any of these evidence sources include non-coding variants from GWAS catalog?
Yep, that's a good question. So again, this is actually, so GWAS catalog is a data source for Open Targets genetics, and Open Targets genetics feeds into the platform. So it's kind of indirectly a data source. And actually you can see it here, for example. So GWAS catalog is one of the main data sources for Open Targets genetics alongside UK Biobank and FinGen, for example. So
So if you're fine, we can take one or two more questions.
Yeah, absolutely, no problem.
Okay, great. Yeah, so someone says that there are 62K targets, but there are only 19K proteins in humans. So how do you explain that?
Yes, that's because some of the targets will be non-protein coding genes, for example, or they might be. We try to have as big as possible definition for each of our entities in order to have as much data as we can. So yeah, they might not all be proteins.
Yeah, and I think even for a single protein, there might be multiple targets.
Absolutely, yeah. Okay, so the next part of this presentation is about target prioritization. So this is a new view that we've introduced in the platform in December. So this is really quite recent. And the idea here is you've found a list of targets. Now, what are you going to do with that? So in this use case where we are a researcher looking at a specific disease, and let's say we want to try and find a target that we want to pursue for a drug discovery program, how are we going to differentiate between the targets? And this is what that looks like. So I will walk you through this as well in the platform itself, but I wanted to show you what this looked like. So again, here I've looked at psoriasis, and I've now clicked over to target prioritization factors. And this is showing me annotation data for the targets that I have listed on the left. And this is separated into different factors, which I will just run through quickly now to give you an overview of how we've sorted it. So there are 4 different kind of target prioritization factors which we've been thinking about. So the first one is precedence. So are there examples of existing therapies for this target? The second is tractability. So let's say you pick a target, can you even modulate it? Do we know that there are binding pockets, for example? Could you create a small molecule for this target? The third one is doability. So do you actually have the tools to study this target? Because do you have ways in which you can test it, in which you can study it? And the fourth one is safety. This is really important because as I mentioned at the start of the presentation, safety and efficacy are some of the biggest reasons why targets will fail. Well, new drugs will fail in the clinic. So safety is a really important question. So will modulating this target result in any side effects? And I've listed some further questions to kind of show you what each of these mean. So for example, in precedence, so do you have any approved drugs or ongoing clinical trials? And our data source for this is Kemble. For tractability, where is the target located? Does it have a ligand? Does it have specific pockets? And here I've listed the data source on the right as well. So we have a specific tractability pipeline for open targets, which we use to kind of inform this section. In the doability section, are there any mouse orthologs? Are there any chemical probes? What tools do we already have that can allow you to find out more about this target? And in safety, will there be any side effects? So is this target under high genetic constraint? Is it in a core essential gene? Is modulating this target associated with adverse events? These are some of the questions that inform this. And then finally, does this target have any paralogs? Is it specific to a target, is the target specific to a tissue? How distributed is it? So these are kind of all the questions that we have when we think about target prioritization. So I'm going to switch back to the platform now so I can show you what this looks like in practice. So this is my psoriasis page. And what I'm going to do here is I'm going to click here, target prioritization factors. So now again, all of the targets on the left are sorted according to the overall association score. So this is the same way in which they are sorted in the target disease association page. But instead of showing you the data sources for associations, we're now showing you data sources for prioritization. And so this is going through all of the different factors of a target, all of the different features of a target that can inform whether or not it might make a good target for drug discovery. And let's say, for example, I'm going to get rid of TNF. Let's say, for example, we look at the example of TYK2. It works in the same way. So we also have a scoring system here, which you can see here. So this is a traffic light system. If it's green, it means that it's the data for this is positive. If it's red, it means it's unfavorable for prioritization. And any prioritization decision is a balance of all of these factors and more. This is, for some reason, taking a really long time to load, so I might try a different one. So here, for example, you have genetic constraint, which is data that we get from Nomad. And this is telling you, for example, TYK2 is under fairly high constraint, which is why this will be here in orange. Other ones that we have, for example, will be tissue specificity. So this is baseline expression from Expression Atlas, HBA, and GTEx. Just showing you all of the RNA and protein expression that we have for this. And again, this is in red because it's quite, it's not necessarily very specific to one tissue. So I'm jumping around a little bit, but it's structured in the same way. So for example, here we have precedence, here we have tractability, doability, and safety. So these are the different factors that I talked about earlier. The other thing I wanted to mention here, so this is all information that we have within our target profile pages, but the way that we look at it here is through the kind of drug discovery lens. So this is a really useful view if you're interested in target prioritization factors. But if you want to see all of this information kind of in one page, you can also click here and go to the profile page for TYK2. So this page is showing you all of the information that I just shown you and some additional widgets which we didn't include. So for example, this TYK2 page will have ProtVista. So ProtVista showing you functional, positional, structural protein information for TYK2. We also have this widget for molecular interactions. So this is showing you physical and functional molecular interactions with TYK2. There's a number of different sources for these because they have shown you different kinds of interactions. For example, here, if we look at intact, you can have a look at all of the different proteins with which TYK2 interacts and explore the publication, so the original data source for these interactions. So this is just some additional data that we have on the target profile page that is not currently in the target prioritization page. And I'm just scrolling down just to show you what that looks like. So most of these widgets will be in the target prioritization page. And finally, I just want to spend a little bit of time on the bibliography widget because it's quite a cool way to explore the literature. So this is showing you, so in this case, this is the profile page for TYK2. So this is showing you all of the publications that we have that are that talk about TYK2 or any of the TYK2 synonyms that we know about. This is quite an interesting addition that we had to the platform where it's showing you similar entities in the literature. So it's suggesting other entities, be they targets, diseases, or drugs that have been mentioned in a similar way in the literature to TYK2. And it just might be interesting for you to see, what other entities are out there that might be interesting to take a look at. So let's say, for example, I was to click on brepocitinib. This is now showing me all of the literature that mentions both TYK2 and brepocitinib. You can see the abstract, for example, and you can see whether the full text is free to read. That's just a little quick stop that I wanted to do on the bibliography widget. I'm going to go back to the page I was on before, so the target prioritization factors page. And I will now ask if there are any questions about this page.
Yes, there are a question about prioritization. Can you rank targets according to one of your 4 prioritization factors?
I don't think you can't, because the score is kind of more of it's more of an indication. So it's much more, as opposed to the association score, which is much more granular. This is very much kind of categorical. It's good, it's not, it's favorable, it's not favorable. So you can't currently rank them according to the individual. Yeah.
Okay. I'm not sure if this question is about the picture which I'm seeing on the screen, but what does the white color indicate?
If it's in here, it means that there's no data for this specific data source for the specific target. And it's the same for the association page. If it's white, it just means there's no data.
Okay, so I think it's from the earlier section, I think. For those uncoding genes, are they accessible on open targets?
Yeah, so if you know, I mean, if it has a target ID, then you can definitely look it up in the platform. I don't have an example off the top of my head, but yeah, you should. If it comes up as a target in your associations page, then it will have a page within the Open Targets platform.
I see that Europe PMC entries associated with a target have a score. How is this calculated?
Yep. So the score is based on how they are mentioned. So the yeah, so for example, the number of times in which they're mentioned in the publication, basically trying to find the most relevant ones for your target. Yeah, I just mentioned very quickly for all of the scoring, we document this in much more detail in the documentation. And it's I recommend you take a look. If you're interested in the scoring, I recommend you take a look at that because yeah, it's much more detailed than I could explain right now in the 15 minutes that we have left.
Okay. So it's completely data organization and correlation, not prediction. So I think the data which you're showing, is it a prediction or
It
mostly
depends on the
correlation?
data source. So yeah, sometimes it will be predicted. So things like I should have off the top of my head, I don't have an example, but sometimes it will be predicted and sometimes it will be curated. It really just depends on the data source.
Are the evidence sources equally evaluated for the overall association score?
So they're not. So I'm going to switch back to the original page just to show you what that looks like. So this is so they have different weights, so they're evaluated differently. So open targets genetics will have a higher weight than, for example, IMPC, which is animal data. You can change this weighting if you'd like to.
So you have weighting profile for targets, I think. So how can I save weighting profile?
Right. So currently you can't save it. I think this is something that we were discussing within the team, but unfortunately, you kind of have to, if you have a specific profile, you sort of have to write it down somewhere or remember it. It's not something that we currently save.
How can I differentiate between strong association and that has not been extensively researched yet?
You can't really. It's kind of, yeah. So I mean, it's you have to look at the evidence. So with all of the evidence that's within the platform, we really encourage you to go and look at the data, to look at the widgets, to look at the original data sources, because ultimately what we're showing you is a summary of what exists. But we definitely encourage you to go look at the original data. So unfortunately, we don't make that evaluation for you. We encourage our users to go and evaluate themselves which associations they think are stronger, even though they might have less evidence. Yeah, just because we don't have a lot of time left, I do want to show you the different ways in which you can access the data. So I'm just going to show you that very quickly. So this is just a quick summary of target prioritization. We've done the walkthrough. So yeah, accessing platform data, and I'm just going to get rid of this a second. So there's different ways in which you can access the data. I've just shown you how you can access it through the web interface. So we've seen that you can search for data for one target, one disease, one drug, one association, and you have the URL there at the bottom. You can also access our data through a GraphQL API. This is really useful if you want to traverse the data. This is the kind of power of GraphQL. You can access different endpoints to retrieve information on associations, evidence, annotation data. So everything that I've shown you in the platform, you can do within the API. You can also go, for example, if you wanted to directly get information for a disease associated with a target and then more information about, for example, let's say more, drugs associated with that target, you could do that within GraphQL, which you can't do within the platform. For this, we would still recommend only kind of looking at one association or one target or one disease, because more than that, it starts getting quite complicated. If you do want to look at multiple targets at the same time, for example, you could use either Google BigQuery or our data download. So Google BigQuery, if you're not familiar, you can use SQL-like queries to access and export all the data to a Google Drive, a Google Cloud bucket, or a local download folder. This is very useful if you have further analyses, for example, that you want to do in Google Cloud. I've put the link here, Open Targets Prod, and you should also get an access to the slides. Everything I'm saying here, you can also find in the documentation. So if you have any questions about this after I'm done, you can take a look at the documentation. The documentation also has a number of examples and things like that to help you get started if this is something that you're not super familiar with. And the final way to access our data is using our data downloads. There's a link to this on the platform itself and also in the documentation. So using the data downloads, you can access JSON and Parquet files, and this means you can really do more complex, more systematic queries. And as someone mentioned, you could use this to do analyses offline, for example. Yes, we also had a question about data licensing, so very important to note. So all of our data and our pipelines are open source and available for academic and commercial use. You can check out our data access and our license documentation in the page that I showed you, so the documentation. I would also highly recommend that you check out the Open Targets community. So this is available at community.opentargets.org. This is our kind of community forum for people to ask questions, for people to share ideas. I use the platform in this way, that sort of thing. We're really interested to see how people use the platform. So if you have any questions or if you've done something cool, please do share it with us. We love to see that. And we also monitor the community. So if you have any questions about how to use the data or what a specific score means or something, we can always explain it to you on the community. That's just a bigger screenshot of the community. And I think that's the last thing I wanted to talk about. I had a quick summary here. But yes, that's the end of my presentation. I did wanted to share a few key links. So if you'd like to, updates from us, you can follow us on social media. We have a newsletter where you can get monthly updates. I've put the key links on the right, so the platform itself. We have a blog where we dive deeper into, different features of the releases. We try to dive deeper into different technical aspects of the platform, the community, as I mentioned, and of course, the documentation. That's it from me. I'm happy to take any remaining questions in the time we have left.
Thank you. Thanks a lot, Helena, for today's webinar, and thanks everyone for joining today. All right, so with that, we'll go back to the Q&A. So one question which is there a standard guideline for determining what specific overall association score qualifies as a disease driver?
Not really. So again, we kind of leave this up to your discretion as a user to kind of to set your own cutoff point, because and all of these scores might change with different releases as we integrate more data and that sort of thing. So no, we don't set a cutoff point for that.
How should targets with favorable targeting factors, yet minimal or no established prior treatment therapies be evaluated in terms of weighting?
Yeah, that's a good question. So, I mean, so for us, these are the kind of scientific prioritization factors. But of course, for example, from a drug discovery perspective, if you're a pharmaceutical company, maybe you probably want to pick a target that doesn't have, previous drugs because you want something new. So that's again kind of a lot of this is up to the user of sort of what are the factors that are most important for you? What do you want to consider? I think in this case, I didn't dig too deep into the data, but I think if you look closer, for example, you can start thinking about what is the direction of effect, what kind of, are there maybe subtypes, are there variants that I can target, that sort of thing. I think that's all more granular data that you can take a look at within the platform. So I think when you look at different targets for evaluation, I would dig deeper into the data. But yeah, I'm not a drug discovery person, so I this is what I know, but I leave it up to the experts.
Great, thank you. One question just came in. How is the new target or new disease updated to the platform?
I'm guessing you mean, for example, if there's a new term or something, or a new target. In that case, that will come through. So for us, when we do a data release, it will depend on what's been added to the different sources that we have. So Ensemble or EFO. For example, if EFO changes the way that they do their ontology, if they have more granular diseases or they reorganize how they do their diseases, that will affect how it looks in the platform. In a similar way, for example, whenever we have a regular release, the status of drugs might change with the Campbell. That will change the way that the drugs appear on the platform, or the data that we have on drugs on the platform, for example. If you do notice things that are missing, or if there's a target that you know about, or disease that you know about that you think is missing, please do let us know. You can let us know in community, and we always have a dialogue with the different providers about this sort of thing.
Next question is about the job opportunity. Are there opportunities to join Open Platform team?
Yes. Oh my gosh, thanks for asking. Yes, we have a jobs page. So we have on our website, opentargets.org. If you're interested in joining us, which is very exciting, you can take a look at the jobs that we have there. If you follow us on social media, I will put when we have new jobs available. So yeah, please do keep an eye out for jobs that we have. I off the top of my head, we are currently recruiting, but I can't remember which ones they are. But yeah, please do take a look at the page, or you can also subscribe to the newsletter. I also put those in there. Oh, very exciting. I hope we do get a new team member.